I'll just introduce him. So this is Professor Tim Mousseau. Tim is a professor of biological sciences at the University of South Carolina. His areas of scientific expertise include the ecological and evolutionary genetics, as well as the effects of Chernobyl and Fukushima nuclear accidents on wildlife. He's published 11 books and more than 200 scientific papers. About 100 of those relate to radiation effects on wildlife. And he's going to talk, maybe discuss, the recent findings he has on germline mutations in bugs and humans.
Thank you so much, David. And funnily enough, Carmel ends with a dog picture. I'm going to start with. Really good to see you. I've seen some of you before, I think, in the past. It's nice to be back in Britain. Although this is Scotland, right? So I'm going to be careful about what I say. I just flew in from Chernobyl. My arms are very sore. In fact, when Jill invited me to come speak at this meeting, Jill and Carmel, my first answer was, I'm in Chernobyl. I'm doing research this month. I've been doing research in Chernobyl for, this is my 19th year, I guess, I've been down there. And we're always there in June, because this is the time when the birds are breeding and the spring flowers are out. And so it's sort of the best time for doing biological studies for the sorts of critters we're working with, at least. And but Carmel and Jill made me an offer I couldn't refuse, and twisted my arm, and so here I am, and it's great to be here. Funnily enough, the main reason for going to Chernobyl this past trip was actually to continue a new study that we started last August. It was sort of one of these adventitious, sort of an accident that we teamed up with a group of veterinarians who were, and NGOs, animal welfare NGOs, who were interested in dealing with the animal control problem around the Chernobyl power plant. They've been given an ultimatum by the Chernobyl authorities that if they did come up with a solution to dealing with all of these dogs wandering about, they were going to disappear in another way. And so folks got together and organized a team of veterinarians from around the world to come in and catch these dogs and spay, neuter, and vaccinate them. And so we saw this as an opportunity to tag on to that project to study a charismatic megafauna, as they call them, a mammal not so different from ourselves, which is wandering around the zone in some of the most contaminated areas. And I brought a team of my 5 students of mine this time. They didn't get to go to the highly contaminated areas. They were stuck in the clinic helping with the dog research, but they did have the experience of a lifetime. And anyway, we've been trying to turn this into something useful and practical for better understanding of research of radiation effects. One of the first things we did was we decided we had to measure the dose that these animals were getting, because this is the #1 criticism we usually get, is that you're just sort of guesstimating the dose that your birds and mice and insects are getting. You need to do it better. I think David said this once or twice. And so one of the things we pioneered last year was the use of very simple dosimeters, putting them on ear tags on the dogs. This year we've also replaced these and put with much smaller ear tags so they don't bother the dogs as much. And I don't know if you want to smell. Can you smell it?
Oh.
And we'll pass it around. That's what happens when you put something on a dog for a year. It comes back pretty doggy. But anyway, last year we actually captured about 400 dogs. This year we managed to catch almost 200. So the program's working, but not completely, as evidenced by these cute little puppies appearing. So anyway, we've been working hard on that project, and that's really kept me from doing a bunch of other things recently. I had promised to talk to you folks about bugs, well, germline mutations in insects and humans. And that's really what excites us at the moment. You know, as an evolutionary geneticist, one of my main ambitions in working in this system has been to document whether or not the radioactive contaminants that we see in places like Chernobyl actually generate perpetual damage, perpetual genetic damage that can be passed from one generation to the next. And for that, it's not as simple as just taking an individual and measuring its genotype in some way and seeing that there's mutations, which is hard enough. It's even harder to demonstrate that the mutations that have been generated, if they are generated, are transmitted from one generation to the next. This is fundamentally an important distinction because obviously the ones that are in the germline that are passed from one generation to the next can accumulate over multiple generations, and especially if we're talking about chronic multi-generational exposure to this kind of low-dose radiation. And you know, one of the questions that's come up from our studies has been whether or not, you know, how do you explain the degree of damage that you see? Apparently, it's much higher than was predicted based on laboratory studies in the past. It's higher than predicted by some of the controlled environmental studies. And so one explanation is that this system is very different. This kind of exposure is very different than what regulators normally engage to assess the risk. They normally do laboratory studies. They normally do look at the lethal dose that kills 50% or has a 50% effect of some sort. And usually these are much higher doses, they're over short periods of time, and they don't look at the chronic multi-generational effects that might be occurring. So, so one of the goals has always been, as an evolutionary biologist, you know, is there an opportunity for this kind of mutation accumulation over multiple generations? And if so, this could actually account for some of the differences in sensitivity that we see versus what might have been predicted by others. And so that's been an aim of our studies. And there's really only a couple of ways to get at this question. And really, you have to look at the genotype of parents and the genotype of their offspring. And so, and you have to then look for what we call de novo mutations, new changes in the DNA that pop up that weren't in either parent. Of course, you have to be sure that the two parents really were the two parents, which is sometimes tricky in human populations as well as others. In human populations, there was a study done 30, 40 years ago in St. Louis that demonstrated that, in fact, the rate of cuckoldry in that human population was as high as 25%, and yeah, it's shocking for some people, not for others. In any case, the other thing you have to be able to do is to look at the genotypes, and you know, for the bugs, one of the reasons we've been working on bugs, firebugs in particular, is that they have visible mutations. The mutations that presumably occur at the level of DNA in some way are expressed in a way that you can see them. And so in the case of firebugs, it's really obvious. Some of you have probably seen the pictures that we've published. They have a sort of a face mask motif, and so if there's a mutation, you know, they'll be missing an eye, or they'll be missing part of their nose or part of their little dimple that they have, and so the human brain is very good at picking up deviations from symmetry, and so it's an excellent model system. And so recently we discovered in Chernobyl that the frequency of these kinds of deviations from perfect symmetry were much higher in the more radioactive areas. And it seemed very clear to us that this reflected some genetic effect, but you have to prove it. You have to use science to test this hypothesis that this is due to genetic mutations induced by the radiation. And so, so with bugs, it's great, because a lot of bugs, especially these firebugs, they feed on seeds of lime trees. And I think you probably have them here in Britain, firebugs. Have you seen them?
We have blowbugs. I don't know if
I'm sure you have firebugs. And I apologize, the file doesn't seem to be on there, but these bugs are highly specialized on this one species of tree, and so they're very local. They don't move. And so this also allows for evolutionary responses and the accumulation of mutations over relatively short periods of time. They get 2 or 3 generations a year, depending on the temperature of the environment. So anyway, we brought these bugs from Chernobyl into the lab and let them do their thing. We actually paired brother and sister through multiple generations in order to concentrate any mutations that might be in this population. And lo and behold, when you mate deformed mom to deformed dad, they have a much higher frequency of deformed kids. And this is consistent. If you have, oh, thank you.
I don't know if you can all see it, guys.
And yeah, I normally bring some posters, but since I've been in Chernobyl for 3.5 weeks before here, I didn't have room. I did bring a gamma spectrometer, so if anybody has a sample they want measured for contamination, we can do that. So the short answer, unpublished at this point, is that the bugs do show heritable genetic variation associated with these deformities that is passed from generation to generation, that actually may account for the fact that these critters seem to show higher rates of sensitivity or higher sensitivity to the contaminants than one might have predicted. But you know, who cares about bugs, right? You know, as an entomologist, I like bugs, but most people really don't care about bugs. What do we care about?
Puppies.
Puppies. You know, the puppy appeal is really strong. You know, I had these 5 students of mine were all young women. They all want to be veterinarians, and you know, and their favorite time of the day was when they got to hold the puppies and you know, rock them to sleep after. Anyway, puppies are great, but what we really care about are ourselves. And so the main goal for the last few years has been to attempt to address this question in a human population. And I actually presented some of this data yesterday, and I was warned not to talk about it in public, especially when there's cameras rolling, but I can give you the gist of it. No, it's fine. I'm just not going to put any pictures up. And the gist of it, this experiment was to do the same sort of thing we've done with bugs, but to step it up to human populations, because that's what we really care about. And there's not any. The advent of modern molecular genetics, you know, the genetics that we can do now, as opposed to just even 5 or 10 years ago, it's an order of magnitude more sophisticated. There's an order of magnitude more information, and it's 2 orders of magnitude cheaper than it was, you know, even 10 years ago. And so, so I teamed up with an old schoolmate of mine who now happens to run a genetics center at McGill University in Montreal, and we got a hold of about 100 families where the father was a liquidator, helped to clean up the mess in Chernobyl, and got a dose. It varied considerably, but from, you know, half a gray to 2 or 3 grays, depending on where they were and that kind of thing. But they actually have done dose reconstruction on most of these liquidators, at least as best they could. And so we have these kinds of folks still around, and many of them have had children. They've gotten married and had children themselves. And many of them live in Kiev, where we hang out. And so we teamed up with the radiation hospital in Kiev to attract these folks to participate in this study by providing free healthcare, essentially. And so we collected DNA from both parents and at least one offspring from about 100 of these families, sequenced their entire genome. And from that now, by comparing, by looking at the number of new mutations, de novo mutations that have occurred between parents and the offspring, we can ask this hypothesis, we can address this hypothesis of whether or not exposure of the father to this serious dose while he was helping to clean up the mess had any influence on the genetic makeup of his offspring, and if so, how much? And there are many challenges to these kinds of studies, technical challenges. Like for instance, I started this study 3 years ago, but it took a year to get the sequences done, and then it took another year for the computer analysis of the data to be generated, and then the postdoc who was in charge of the analysis got a real job and had a baby, and so anyway, so it hasn't been published, which is why I can't show you the actual results. But the basic bottom line is that it looks like 70 or 80% increase in this de novo mutation rate, which could, so almost a doubling, and so it could in part at least account for some of the other kinds of consequences, health consequences that are being seen in this population. Although, again, with humans in particular, it's very complicated and very difficult to dissect out all the extraneous confounders or potential confounders that might be influencing these populations. But that's really been our main goal in the last couple of years, is to get a handle on this really fundamentally important question of whether or not germline mutation rates increase, and whether or not they can accumulate over multiple generations, and whether they might be associated with some of the diseases and other abnormalities, developmental abnormalities that we see in these populations, which include, smaller brain sizes in these children, lower cognitive function in these children, higher rates of cataract in the eyes, a variety of different endpoints that aren't just cancer, which tends to be the focus of these kinds of studies, but also looking at these other biological effects, which we can also study in animal populations. So that was the intent of my presentation, was to cover that, and I guess I've managed to do most of that. Any questions on that part of? Yes.
I just don't want to monopolize the conversation, but actually there was a group of men who, on April the 28th, were exposed in 1958 to the grapple Y nuclear test in the British Royal tests in the Pacific. Now, what's significant about grapple Y is it was the only nuclear test conducted by the British in the rainy season. And when we looked at the epidemiological data, we found that a very high proportion of things like skin cancer, skin disorders, cancers, and also family problems, we call them fertility problems, were associated with that particular test compared with nuclear test veterans exposed to other tests, because this is where they were subject to radioactive rainfall, which we know, as with Belarus and so on, that there's a much bigger level of exposure. We then looked at the grandchildren, and we found that whereas there was factor of four joining, as it were, with the joining the Test Veteran Association, associated with this particular grapple Y. When we came to the grandchildren, the ratio was 8:1 and not 4:1, which is almost a doubling again. Now, I'm sorry about putting a plug in there, but I have actually got a publication here that actually goes into that in great detail, so if anybody wants to consult me afterwards. But the point is that these were 3,700 men. I'm not trying to say, well, we've got more people than you have, but what I'm trying to say is it wasn't as well documented. But the key point was that it was a double-blind experiment, because the men who joined the association and the people who ran the nuclear test had no idea that that particular test was actually, in retrospect, much more dangerous. Whereas, of course, with Chernobyl, the problem there is it isn't a double-blind test. But with grapple-wire, it was a double-blind test, and we got this very phenomenal change in the number of people with what you might call adverse complaint effects. Now, I'm just saying that totally backs up what you're saying. You're getting an 80% increase from generation to generation. Well, we were getting, obviously, on smaller numbers in terms of actual birth defects reported, but it was a 4-to-1 in the first generation of children, and then it was 8-to-1 in the second one.
You know, the more I learn about that era and what was being done around the world, especially related to atomic bomb testing, the more I worry about the current world situation. Because people are clearly capable of anything, including experimenting on themselves, and it's it's kind of a scary thing. We hope to get to the Marshall Islands to do some more critter work as well. I think the more we look at these other scenarios, these other environments, and try to replicate these kinds of studies in under different conditions, the more we learn about the actual mechanisms. You know, when this meeting we had earlier this week, you know, there were two main themes that came out of the meeting. Well, for me, there were many more, but two that struck me as being particularly interesting were the fact that more and more we're recognizing the importance of complex interactions among multiple stressors as driving potentially synergistic interactions and leading to much larger impacts in some circumstances than predicted. Chernobyl had a lot of heavy metals associated with it, and one of the things we're doing with the dogs is we're actually analyzing all the hair for heavy metals as well. The dogs also, you know, it's a fairly small population, so we're going to genotype them all and recreate the pedigree so we'll know who mom and dad are. We won't, you know, dogs are, you know, amazingly promiscuous. That often there's multiple fathers and a bunch of puppies. So, but anyway, that's another issue. Same with cats. But yeah, I think we're starting to develop the tools that allow us to rigorously dissect these kinds of issues. Yes, ma'am.
The thing about the liquidators is you're doing the ones that had survived. I mean, there's a huge number that have survived, and you don't know what the ones that have died before us, what happened to their children, do you? And that was probably a bigger multiplier than the one you're using now.
You know, yeah, I think you're right. And that sort of applies universally for all of these kinds of studies. You know, our bird studies, you know, we don't get the ones that died. You know, we only get the ones that survived. And so what this means is that our estimates of effect are likely to be very conservative. And you know, the true effects under perfect conditions, if we could have preserved everything, would have been even larger. And we had this discussion about cancer in natural populations. You know, there's a greater recognition now, as more emphasis has gone into this, that cancers are actually found in natural populations of birds and other critters, and we just haven't looked. And we started looking for tumors, not necessarily cancers, but tumors in the birds, and found that 1.5% of the birds in the most contaminated parts of Chernobyl were showing tumors that just weren't seen anywhere else. That doesn't sound like a lot, but compared to, you know, what's the normal occurrence, it's huge. And the fact that a bird with a tumor or a cataract is unlikely to survive particularly well, that means that it's probably a lot more. So yeah, no, that's a really important point.
Can I just ask, are the dogs always adverse?
Yeah, so it depends on what you're looking for. So, you know, one of the first applications of radiation was to zap a whole bunch of seeds and look for mutant plant types that were suitable for agricultural use. I think in nature, my suspicion is that you know, mutations are always happening.
Exactly.
There's constantly a reshuffling of the genome, and you know, we've been here. You know, biological critters have been on the planet for 4 billion years. We've essentially had the same, you know, system of inheritance, and so, in all likelihood, all of the possible permutations have been tried a few times, more than likely, more than a few times, and so any changes that occur at the genetic level, at least, are likely to be either neutral because of the redundancy in the genetic system, or or slightly deleterious, because if they were really deleterious, they'd be eliminated fairly quickly. And beneficial mutations are new, de novo, novel beneficial mutations are expected to be extremely rare. But again, if the environment changes a whole lot, then there may be opportunities for that to change. So as an evolutionary biologist, my expectation is that the mutations of this sort are always going to be deleterious or neutral. What else do I want to talk about? The second key point to come out of this meeting earlier this week was that there seems to be a consensus, and just stop me when you need to, there seems to be a consensus now building among the radioecology community that a really valuable endpoint for a target for study is this question of biodiversity, or abundance and distribution of organisms. There's many ways to characterize this, and we had a lot of discussions about different mathematical tools that might be used to assess biodiversity, and that discussion is ongoing with respect to a lot of other issues like global climate change, etc. But just recently that the radioecology community seems to be embracing that kind of concept as a valuable endpoint for assessment of impact. I was really happy to see this switch, since we've been working on biodiversity for the last 10 years as a primary endpoint to our work, and I don't think, so basically distribution abundance as endpoints for ecological risk assessment, and as I would say, it's biodiversity that counts, stupid. There are challenges to doing that as well, to working in that domain. But the main challenge is having people who know the natural history, that know the critters, that can go out and do the inventories that are needed to really assess how biodiversity, distribution, and abundance of organisms might be changing in response to environmental change. And the way we've done it has been very simple, and we have a lot of papers on the topic, and happy to share them with you. But it's been simply to go do these biotic inventories of all the birds, all the bugs, all the critters we can find at hundreds of locations in both Chernobyl and Fukushima, and to monitor how they change over time. Chernobyl, we've been doing this for 10 years on and off, in Fukushima since 2011, with the exception of 1 year, we didn't get there. And the overwhelming conclusion is that there are major, highly significant declines in both the average abundance and average functional diversity in the more radioactive areas, and that's after removing the effects of other important habitat variables like water, soil, vegetation, and statistically focusing on the radiation effects. One of the reasons you have to look at so many different locations is to get at that, to be able to treat that issue statistically, to remove the effects of other variables that are clearly even more important than radiation in most circumstances. I did want to show you a map, but I don't have it here, and that was a map of the Chernobyl exclusion zone, and but I'm sure most of you in this room have seen them. They, when you turn on the TV around in April, you'll undoubtedly find some television show going on about how wonderful Chernobyl is for wildlife. The animals are thriving, they've exploded in numbers. And like so many of these kinds of presentations, there's some truth to that. The fact is, Chernobyl is an enclosed area. It's a wildlife refuge. It's got a fence around it. People aren't allowed to come in and go hunting. And so as soon as you remove that human pressure, populations rebound. Darwin very carefully explored this issue. We'd be up to our armpits in elephants everywhere if they weren't being culled irregularly. Within 1,000 years, there'd be more than 1 billion elephants on the planet if they weren't dying for one reason or another. And so, the same's true for everything, everywhere. In Chernobyl, the question really is: are the rates of increase similar or different to other wildlife refuges in Europe. And the other question is whether or not, and the other aspect about Chernobyl that I didn't mention is that most of it's actually clean. Most of it's actually not contaminated significantly. There's only a very limited area of Chernobyl that's highly contaminated. So the question is there a difference in animal performance in radioactive areas versus the clean areas within this Chernobyl zone? And then even more interestingly is, do you see the same kinds of patterns in other parts of the world where there's radioactive contamination like Fukushima? Oh, I'm getting the time to go. It's okay. So anyway, the bottom line is that a lot more work needs to be done to characterize these kinds of patterns of decline in a way that might be useful for the regulatory agencies to employ in risk assessments, especially if they want to apply it towards humans. But I'm optimistic that we're on our way towards that, and I think there'll be some valuable information coming in the next decade or so. Well, that's all I got to say. Without slides.